Hello friends, welcome back to my YouTube channel Nestin Padasalai. In this video, I'm going to discuss 100 topic with regarding to GATE Biotechnology examination. Say if you're already completed your GATE exam preparation, you can use this particular video as a checklist in case if you miss any one of the topic from the 100 topic listed over in this video, you can study them. Or else, many group of students will be preparing for GATE examination only for 1 month. And February 4th, our GATE exam is there. Okay. So, by using this video, if you study only this 100 topics which are highly important, you can definitely clear your GATE examination. Okay. So, for two group of students, this video will be really beneficial. So, without any delay, let's get into the video. So the first important topic itself, you should know about all biochemical pathway and the location of those biochemical pathway. The second topic is the antibiotics and their targets. So definitely at least one questions will be coming from this particular antibiotics. Next thing, you should know all types of RNA and the function that they invert. And next fourth topic is viral classification and the family of virus. At the same time, we should also know about this new virus called coronavirus. So, since the virus is getting back, at least one questions you can expect from that particular virus. Next, you should know about the chromosomal disorder. You should know the cause of the chromosomal disorder and how it will be represented and the karyotype. Say if you are studying Down syndrome means you should know the karyotype, that is 47 XXX plus 21. There will be extra 21st chromosome. So you should also study the karyotype and how it is represented. So Down syndrome is called as trisomy 21. So you should study in this particular manner. Next thing is regarding to inborn error of amino acid metabolism. So you should know the enzyme which is lacking in that particular disorder. Next, you should study about the energetics of biochemical pathway. How many ATP is produced? Say if you are studying glycolysis means how many ATP will be produced during anaerobic pathway and aerobic pathway. Like that you should study. Next thing, you should study electron transport chain, and while studying you should also study about the inhibitors of electron transport chain. Next, you should study about restriction enzyme. There are three types of restriction enzyme, type 1, type 2, type 3. You should know about their recognition site structure. In addition to that, you should also know about blunt end enzyme and the enzyme which will be giving sticky end. Next, study about cancer, and regarding to biochemistry, you should definitely know about the enzyme inhibition. Okay, LV plot, everything. The graph also you should know. Okay, next equation. So you should know about the mutation, types of mutation, and also you should know about mutagen. Next, extra chromosomal inheritance, and you should know about transposon and transposable elements like example of class 1 transposon, example of class 2 transposon, like that you should know. Next, know about this Ames test, Leventhal paradox, genetic code, and also know about Wobble hypothesis and ABO blood grouping types, everything, and also vitamins. So some vitamins are used as a cofactor in different enzymatic reactions. You should know about those things. Next, glycosidic linkages, epimer, anomer, like the basic information. So when you are studying carbohydrate, you should know about these kind of things. Okay. Next, entire detail about amino acid, the pK of amino acid, the structure of amino acid, each and everything you should know about amino acid. Which amino acid will be present in the interior of protein, which amino acids will be present in the structure of protein, like that you should know. And you should also know about the microorganisms that are used in the industrial sector. Like which microorganism is used for production of vinegar, like that you should know. Next, vector used in gene manipulation, and regarding to immunology, you should know about major histocompatibility complex and complement pathway, complement protein, vaccines, and while studying vaccine, you should also have an idea about corona vaccines, okay, and immunization, types of immunization, and next antigen processing and presentation and nitrogen fixation, and you should also know about monosomy, disomy, nullisomy, trisomy. You should also know about the type of aneuploid and the aneuploid number, how nullisomy will be represented, like that you should know about the representative numbers, okay. Next, you should study about immune cells with special focus to B and T cells. Okay, so you should know about complete information about B cells and T cells. In addition to that, you should also know about other immune cells like macrophages, NK cell, like that. Next, gene transfer techniques and reporter, gene selectable marker, While studying the gene transfer technique, definitely you should know about TI plasmid. So while studying TI plasmid, you should know about where genes. Okay. Next, enzymes used in recombinant DNA technology and reagent with a preferred cleavage site. Okay, so you should know some chemicals are used for enzymatic cleavage. Like if you take trypsin, trypsin have a specifically cleave in the C-terminal side chain of lysine and arginine. Same if you take cyanogen bromide, it will be also cleaving at methionine side. So you should know about the reagent and the preferred cleavage site. Next, you should know about the location of mitochondrial enzyme. Some enzymes will be located in the interior of mitochondria, outer membrane of mitochondria. Some will be located in the inner membrane. So you should know all detail. Next, translation inhibitor, transgenic animal and transgenic plant. Everything you should know. Okay. Next, you should know about the plant natural product, like what is a kind of, whether it is an alkaloid or tannin or lignin, and from which part of the plant that particular product is being produced, and what is the application. Same with studying secondary metabolite also, you should know about entire thing. And next, you should know about indicator organism, and plastic transformation is highly important topic. So please don't try to skip this particular topic. Next, how knockout and knock-in is animal is performed. Next, CRISPR-Cas technology. Then you should know about site-directed mutagenesis, RAPD, RFLP. Next, you should know about the microscopy. So you should know about each and every microscopy, like what is the purpose of dark field microscope, then under when confocal microscopy is used, and you should also know about few equations related to microscopy. Next, you should know about chip sequencing, microarray. So you should also know about the spectroscopy. Then UV visible is used, when IR is used, when NMR is used, when CD is used. Okay, after studying these topics, you should also have an idea about DNA footprinting, DNA fingerprinting, and SDS page E2 hybrid system. Next, you should know about thermostable DNA polymerase. So till now, we'll be having only tag as a thermostable DNA polymerase. Then there were some other thermostable DNA polymerase scientists have found. So you should also read about those things. Next, have a basic idea about PCR, the principle, the types, then gel retardation assay and chromatography. So while studying chromatography, you should also study about the elution condition. The elution condition just tend to vary. Like if you take affinity chromatography, the elution condition is that changing pH or polarity or ionic structure. If you take hydrophobic interaction chromatography, the elution condition get varies. So you should know about the elution condition used for different chromatographic technique. Next thing you should know about hybridoma technology, expression vector, biofilm, staining technique used in microscopy. While studying staining technique, you should also study about gram-positive bacteria, gram-negative bacteria. And study about Hollandric gene, enzymatic type of enzymatic reaction, like when decarboxylation is used, when oxidation is used, when transamination is used, like that you should know. Next, you should know about puromycin, autoimmune disorder. Differential centrifugation is highly important topic. So knew about the centrifugal speed, centrifugal time, and at that time what part. So generally differential centrifugation is generally used to separate organelle and subcellular component. Okay, so study about them. Next, study about pedigree, action potential, ABC transporter, and substrate level phosphorylation. So while studying Krebs cycle, you should know about SLP and which enzyme will be catalyzing the steps. Next, know about the type of microbial medium. Have a brief knowledge about McConkey agar, EMB agar. Okay, sorry. Next, know about the major nutritional modes of bacteria, microbial growth, antimicrobial resistance, structure of antibody, and types of antibody and function. You should know about IgA, IgE, IgD, everything you should know. At the same time, you should also know about Fabry fragment of antibody. Next, know about hypersensitivity reaction, horizontal gene transfer. So in evolution, you should know about adaptive evolution, speciation, selection, genetic, the founders effect, the basic evolutionary concept. Next, know about this ECM, cell cycle and control of cell cycle, prokaryotic cell structure and function, eukaryotic cell structure and function. And you should also know about this comparison of three domains of life. Like you should know about bacteria, archaea, eukarya. And regarding to this industrial biotechnology or bioprocess engineering, even though you can't able to cover entire topic, since if you are a MSc biotechnology, you could be able to understand the trouble which we will be facing. Know about the degree of reduction, the formula used for calculating degree of reduction, theoretical oxygen demand, what is meant by Newtonian fluid, what is meant by non-Newtonian fluid, know about the bioreactors, and same time you should know about principle of chromatography. And after that, you should know about plant growth regulator and elicitor and protoplast and what is meant by protoplast fusion, what is meant by somatic hybrid and hybrid. And you should also know about types of RNA polymerases and study about operon, tripoperon, lacoperon, arabinosaperon. Next, study about the structure of DNA, tRNA, messenger RNA. At last, you should also study about virus associated with the human cancer. So these are the list of important topics. These are the 100 important topics. Definitely questions will be coming from this topics only. So please speed up your preparation and use this topic. You can either note the topic in your preparation notebook, and if you start today, you can definitely clear this GATE examination. All the best for you, friends. If you still want more notes, then please follow my channel, subscribe to my channel. Thank you, friends. Thanks for watching this video.